3.2, yes.
Ow!
Yes.
I just broke my will. I'm in the middle of class.
Okay, we're on page 546. My turn. Page 546, we're studying the animal-like protists today, and some of them you've been looking at in the microscope. The first phylum, and the animal-like protists are classified based on how they move. The first phylum is called Coleophora, and you're going to have to memorize all these phylums. They will be on the next test. So, there are 4 phylums of... I'll write them down as we go. We're in Kingdom Protista. The first phylum is Coleophora. Coleophora.
Bro, these are different colors.
And those are your ciliates. And when I say the ciliates, I mean organisms that move around with little hairs. Their whole body is covered in little hairs. And they use these hairs to move around. The hairs are called cilia. There's 7,000 different species of them.
Wow.
We have one in there, actually 2 types of paramecia in there. Paramecia are ciliates. Their body is covered with little hairs, and each little hair goes like this, and it helps them move. You saw these under the microscope. Some of you have seen them already. Cilia on the paramecium are used for moving and feeding. They can pull in food with their cilia. They also have contractile vacuoles.
Is that where like it sucks in and shoots it out?
It collects water and then shoots it out. We've already talked about those.
Everything looks
This is
on here.
what the body of one of them looks like. And there's the little hairs all surrounding its body. They have 2 nuclei. A macronucleus runs most of the functions of the organism. And the micronucleus runs the reproduction. And here it shows you a couple of them reproducing with one another. Paramecia sex. And then they all have this oral groove, which you can see under the microscope if you can look at these close enough, where food is sucked into the oral groove, and then food is taken down this passageway called the gullet, and then forms a vesicle around it. And you can see a bunch of these food vacuoles floating around inside the cell. There's the contractile vacuole. And then once the food has all been digested, the food is released out the anal pore. Yeah, anal pore.
I got one of those.
Watch the video footage and see if this is what it looks like under your microscope. Oh, it's not working.
Enable all.
I like how you tricked those in the video
In
here today.
paramecium, specialized structures called contractile vacuoles.
Right, please.
Excess water entering the cell is expelled through a tiny pore, the contractile vacuole pore, formed when a vacuole contracts, discharging its contents, only to re-pore again. To study feeding behavior, these paramecia were offered red Kerman particles. As they move, the cilia create invisible whirlpools that entrap the particles and whisk them toward the mouth opening. The particles are concentrated inside the animal to be digested by acidic enzymes in the food vacuoles. Nothing is entirely edible, even for protists. They rid themselves of waste products through a slit in the cell membrane called the anal pore. The remains of undigested food can be seen extruding from the paramecium.
Ew. Let's go into the bathroom.
That's poopy.
Good job, Tucker.
One more video. One more paramecium video. Doesn't seem to be working. Oh no, I don't know what that is. Here's 2 paramecia conjugating. Conjugation means having sex.
I thought they have 2 nucleuses. Where's the micronuclei?
Good question. Here's another type. Listen. Here's another type of cilia called a stentor, which looks like a big trumpet, which has cilia at the base of the trumpet.
I would go more along the lines of a flute.
Locomotion, that is the ability to move about, is very highly developed in protists.
Clarinet.
Protists basically use any one of 3 different cell structures for locomotion. Perhaps the most common of these are the wiggling hair-like structures called cilia, seen here. Organisms possessing cilia are classified into the protist phylum known as the ciliates. Besides being used for locomotion, many ciliates also use cilia to sweep food into mouth-like openings in their cells.
Those were stentor at the end there.
Or like a 1 second thing of stentor.
Yeah, those little things.
It's like 2 or 3.
Keep in mind, nobody even knew these things existed before the microscope was invented. The guy that first looked at pond water under a microscope and saw all those little creatures, he freaked out.
Well, sure you
Well, I mean, I wouldn't really
He's
care.
told everybody there's little animals everywhere and nobody knows about them. But
No little animals everywhere, guys.
and they're like, what's wrong with this guy? He's like, I'm telling you, you gotta look.
What?
They're everywhere.
Now, the next phylum is Sarcodina, and we have a bunch of these too. By the way, the Euplodes, I don't know if you remember looking at Euplodes, that was a Ciliophora. The two, we have a couple of Paramecia in there, those are Ciliophora. It says on your handout that has all the organisms, can I borrow this for a second? It says the phylums next to the names. So we have Blepharisma with Ciliophora.
Bro, is this the essay?
I can't remember what the essay is for this chapter.
It's so great.
Now, Sarcodina are your amoeboids. Okay, my turn. The faster we get through this, the sooner we get to go back in the lab. So if you'd stop talking, that would really
Yeah,
help.
just shut up, man. Please. Thank you. Got you, bro.
Quiet, please. So, the amoebas, there's different types. You have your regular amoebas and your shelled amoebas. And these are your Sarcodine is the name of the phylum. And they all use what's called pseudopods. Pseudopods are extensions that look like little arms that can extend from the body and wrap around things. Right. Here you have an amoeba wrapping around a little organism and engulfing it into a vacuole. And that's what these things do. They gloop around and engulf things and eat them. They also have contractile vacuoles that squirt out water. And here's one up close.
An amoeba moves by means of cytoplasmic streaming. But that's not all. Amoebas use cytoplasmic streaming to engulf prey. In this case, an unfortunate paramecium.
See, it caught a paramecium there. Look how big the amoeba is. Amoebas are big.
Bro, who was the first one to find it in this class?
I found a dead one.
I found an alive one. Well, guess what?
I found everything.
Watch this. I like this video. Watch the little organism barely escape. Watch this little organism barely escapes. Watch this.
Amoebas use cytoplasmic streaming to engulf prey.
Stop! Yeah! Shut away,
An
see?
unfortunate paramecium.
Suck it, amoeba.
One more time. I love how it barely gets away.
Watch it again. It's like a movie.
It's like the end of an action movie.
Amoebas use cytoplasmic streaming to engulf prey. In this case,
Rewind it. Do it again for one time.
What
One time,
happened
bro.
if it didn't escape?
It would get eaten.
Sports car.
What is that?
It's like a 50-50 chance of living it out. You escape it. They'd have like a tea party. And then they would
Organisms.
Quiet, please. Stop talking.
Protist phylum called the rhizopods.
They're actually called sarcodines now. They used to be called rhizopods.
Why'd they change?
These creatures move by extending projections of their cytoplasm known as pseudopods, or false feet, and then pulling themselves along. This unique style of locomotion is called amoeboid movement.
That's what the amoebas did.
And all this material inside the cells here, it's neat to look at these under the microscope. All this stuff, those are the little organelles that we study. The mitochondria and the endoplasmic reticulum and all that stuff. And you can see it actually moving along inside the cells. But these are real good creatures to study cells with.
Can we
boring.
see that?
Yeah, you can see it just that big.
Number one, yeah.
Yeah, number one. Now you have to use, because these organisms are so big, you have to use what's called a depression slide. It's a slide with a little dip in it, so that when you put the cover slip on, you don't crush the amoeba.
Oh.
So if you want to look at number one, or also Volvox,
17.
which is 17, you got to use a slide with a dip in it. We have them out there. They're called depression slides. They're in a little case on the shelf that's called, it says depression slides.
Cool. Yeah, I think I actually.
Now, some of the amoebas have shells. The shells are called tests. And amoebas with shells are called radiolarians. They're also called foraminiferans. And we have two types of shelled amoebas in there. They're number 2 and number 3. And you can see the little shells. They're brown. They look like brown circles. But if you had an electron microscope, you could see them. They look like this.
Are they really?
Yeah.
No, he just said that.
The ones we look at, this is exactly what the ones we have in our microscopes look like this right here.
Do you have the light?
They didn't look
I don't have the light behind them making it shine. But
that would be cool.
the bodies of these things, when these things in the ocean die, they float to the bottom of the ocean. And over time, their bodies build up, and that's what causes the white cliffs of Dover in England to be white, are the bodies, the shells of these foraminiferans. Millions and millions of foraminiferan shells.
Billions.
Billions. And it makes the whole cliffs white. See those people down there? Look how big the white cliffs are. So those are your amoeboids. And then you have your phylum Apicomplexa. Those are your sporozoans. Sporozoans reproduce with spores. They're all single-celled individuals that reproduce with spores. Often they're parasitic and cause disease. And we're interested in these organisms because they cause the worst disease in the world, which is malaria. Malaria kills the most people of any disease. And no, it's carried by a mosquito called the Anopheles mosquito. And if the mosquito stings you, it injects the spores into your body. Now, luckily, we don't have it here, but they have it in Africa and Asia and South America, and it really causes problems. The little spores get into your body and go to your liver, where they reproduce. They then produce more spores, which go to your blood cells and rupture the blood cells. And it causes you to have really bad fever and really bad sickness, and it kills something like 3 or 4 million people a year.
Wow.
Do they have like any way to like cure it, or?
They have medication, but the malaria bugs are becoming resistant to the medication through evolution. They're evolving resistances. There are some types of malaria that are resistant to all medications, and it often kills the person who gets it.
So there's no vaccine?
For those of us who live in
Developing
northern
one right
climates,
now.
mosquitoes go hand in hand with summer. We've all been bitten, but the itchy red bumps go away in a few days, and we forget all about them. Until the next time we're bitten. Mosquitoes themselves are fairly harmless, but can transmit many diseases, including malaria, through their bite. In tropical climates, malaria is a plague. The cause of malaria is a protozoan of the genus Plasmodium, a single-celled organism with a nucleus. It is spread from one infected person to another through the bite of the female Anopheles mosquito. When the mosquito feeds on the blood of an infected person, it ingests the Plasmodium, which lodges in its gut and produces spores. The infected mosquito transmits those spores through its saliva, infecting each person it bites. Once bitten, the Plasmodium parasites multiply in the victim's blood. Then they travel to the liver, invade the cells, and multiply again. After 1 or 2 weeks, the parasites penetrate the red blood cells, where they multiply yet again, gradually breaking down the cells. The disease works in cycles, making a person ill with high fever and intense paroxysms or tremors for several days. Then for a short while, the person feels better before the cycle starts all over again. Left untreated, the Plasmodium reaches such high numbers and inflicts such great damage to the blood and internal organs that the body is unable to fight. Many who contract the disease will die from it. Researcher Robert Hancock explains.
You may suffer these paroxysms for upwards of 30 hours. And if that's not enough, your body will have another day to recover, and then another cycle will start. And the people who die of this disease die of sheer exhaustion.
Malaria is now found in most tropical regions of the world, but the continent of Africa is hardest hit. Close to 90% of the world's malaria cases occur in Africa. Worldwide, more than 120 million people each year contract malaria, and the disease is responsible for more than 1 million deaths. The push to slow the spread and ultimately wipe out this deadly disease is a race against time. Thousands of people are infected with malaria every day. Scientists predict that if the global warming trend continues and climates get milder, the dreaded malaria may invade our own country in the not-so-distant future.
Move to Alaska.
Let's think about that.
One reason we don't have malaria is too cool up here for that mosquito. The mosquito only likes warm weather. So we're kind of lucky there. The global warming, the mosquito is moving up into northern parts of Mexico. We're scared it's going to keep coming up. So anyway, malaria is a terrible thing. They are developing, scientists are trying to develop a vaccine where you can take it, and if you take the shot, you'll never get malaria. And they're coming close to developing that. If they get that, that'll save millions of lives. Yeah.
In Texas,
In the world,
they had
they're popular.
mosquitoes too, but are those like the same kind?
Different kind. Yeah, the Anopheles mosquito is a really big mosquito.
Well, because like they're talking about the
Like
news.
Jumanji size mosquito?
Yeah, big.
Oh,
That's like tennis.
that's like my worst nightmare.
tennis racket size.
Yeah, that's freaking scary.
They
All
have
right, the
some
final
right
phylum
now,
is
but-
Zoomastagina, which are called zooflagellates. And these things are little bugs. I'll show you some pictures of them. They're little protists that, there's some. They have flagella, that's why they're called zooflagellates. They all have flagella. There's the flagella, the tail. They're real small. These ones invade the human bloodstream and eat up the blood vessels.
Those look like apricots, the little dried apricots you get.
Yeah, they kind of look like blood vessels.
These are blood cells, and these are the little flagellate organisms.
They look like blood cells.
And if you have these, they're called trypanosomes. You have a disease called African sleeping sickness. And it makes you sleepy all the time because it eats up your blood cells. And you don't get enough oxygen to your brain and you're sleepy all the time. There's a different type of sickness called American sleeping sickness that's not in North America, it's in Central and South America. But it's caused by a different flagellate protist. We have one of those flagellates here. It's not a disease-causing flagellate.
Is it like
Pandorina, number 6, is a flagellate. So, yeah, I'll answer your question. Numbers 1 through 9 are all protozoans from these phylums, these 4 phylums we've done today, except for number 5. Number 5 is a euglenophile. We'll talk about that. Question?
Are there flagellates? Is that what they're called?
Flagellates, yeah.
Are they? Do you get them in your body when you have mono? Does that make sense?
These are injected by a fly called the tsetse fly. You ever heard of the tsetse fly?
Yeah. Oh yeah.
It's a biting fly that can inject them. I don't know if you can get them from any other sickness. Mononucleosis is not that.
Number 8, did you even eye out? Or?
What's number 8 say?
Phylum Silophora, but there's a
Coleophora. Yeah, that's this one. I might have misspelled it.
Okay.
Here's video footage of one of these things.
Video footage!
And then we'll go back to the lab.
Ooh, baby.
Oh, I should like one.
The flagellate protozoans, also known as mastigophorans, move by means of flagella. In many protozoans, the flagellum may either pull or push the cell, depending on whether an undulation begins at the tip or the base.
Some of those flagella pull the organisms along. Okay, so I'll let you get back to it, and make sure you clean up after you're done, and have a good weekend. Hopefully we can at least do one or